So chapter 15, 16, 17 activity sheet. We are trying to put together some correct answers. Most of the time, they will be giving the answers. And if needed, I will explain. And that is how the activity sheet filling goes. And don't consider this as a video that gives you answers for all the activity sheet. What I mean to say is, just because you're listening to the video and blank needs to be filled with this word has been said doesn't mean that's the right answer. You have to confirm the answer. You have to learn the answer. You have to learn the material on your own and also make sure that what has been given is also correct. If you have a different answer, then message me, whoever is listening to the recording, as well as the ones who are online. Okay. Brittany, Tracy, you guys said you have the answers. Stacy, Aminda, if you also have the answers, make sure that you speak and give the answers, okay? Tracy, can I go with you?
Yes.
First question, read the blank and also give your answers. Go ahead.
Okay, yes. Number one, chromosomes, chromatin are made of the protein and the DNA.
Good.
Okay, and the second, genes are located on chromosomes and are made of DNA.
Very good.
Third one, P is the model organism Mendel used, whereas Morgan did his experiments with fruit fly model organism.
Yeah, green peas, they call it green peas.
Oh, green pea, okay. Number four, Morgan's monohybrid cross answered, white-eyed allele must be recessive. I was hesitating for this one.
Yeah, Morgan's monohybrid cross actually answered that the genes are on chromosome.
Oh.
Okay, next.
Number five, Morgan determined that the white-eye mutant alleles must be on the X chromosome, which is true.
Good.
Number six, sex-linked gene is located on X or Y chromosome.
Simple, right? We have been talking about it from mitosis chapter. Next.
Number eight, sex-linked. Number seven. Linked genes are two genes on same chromosome, which is true.
Good.
Number eight, sex-linked inheritance cross. Parents have normal vision, but their son is colorblind. Where did he get the colorblind alleles from? The answer is from both parents because there are both recessive gene carriers.
No, it's a son. Son always gets his X chromosome from mother. Why did I say X chromosome here in case of color blindness? Because a normal vision mutated version is color blind version and it is on the X chromosome. And
I
son
see.
gets it from mother, so mother has given it.
Yes. You're right. It's sex-linked. Got it.
Oh, okay. By the way, again, linked genes could be on the autosome, like chromosome 1, 2, 3 to 22. They are numbered. And linked genes could be on the sex chromosome. Linked genes are any two genes or all the genes which are on the same chromosome are all called linked genes because they transfer as a package. Like, for example, generally, blue eye with blonde hair color, eye color and hair color, they are on the same chromosome, they say. Genes are on the same chromosome, they say. Whereas, can it happen that people have a blue eye color with dark hair? Yes, that is due to crossing over, they say. But then later on, studies have shown that actually both eye color and hair color and skin color, they are all actually same. But we do not know for sure. Science has been giving that earlier, so we will go by that for now. You can continue if you like to. Question number 9.
Okay, number 9, what is Barr body? Who has it and where? The inactive X in each cell of a female condenses into a compact object called a Barr body, and the female cell has it. And
Continue.
Yes, female cells in female has it, and where it's inside the nuclear, and it's on X chromosome.
It is the entire X chromosome. So females have 2 X chromosomes, right? So this entire X chromosome curls up or is tightly knotted with proteins, and so that its genes are never accessible. So in all of us, in all cells, we have a Barr body, which is one X chromosome which is completely condensed. In all of us, we have it. Cats have it. Dogs have it, because all these organisms, females have XX chromosome. But there are other animals which do not have XX in female. For example, in honeybees, it's not the story. Grasshoppers, it's not the story. So there are many organisms like that, and that comes in exceptions later. So just want to let you know that this is in all cells, whereas in ovary cells, this gets uncondensed, it seems. Where it was condensed, but then it gets uncondensed so that the genes can be used. So in female body, all cells, not in ovary cells, not ovary cells I'm talking about. Continue, Tracy. Otherwise, if you have anything else, add that as well, otherwise.
So Professor, I just want to confirm, so you said the Barr body is entire X chromosome?
Exactly. This
It's
is
not a
2
one
X chromosomes.
Oh.
Entire X chromosome gets so condensed tightly that none of its genes are accessible, because we don't need double, right? We need only one X chromosome.
I see.
In all our cells.
Okay.
But then what do they have to do with my skin cells or pancreas? It seems X chromosome is not all about being a female or not being a female. X chromosome has normal vision, right? If you have normal vision, then you don't need 2 normal vision genes. One is enough. That's why the other one goes.
But it doesn't have to be the same alleles. One can be normal vision, the other one can be abnormal. So which one is inactive and the other one will just show up. Okay.
Yeah.
Okay.
Yeah. I'm remembering 10. Something, but that will be too much for you if I say that. So
I understand. For my level now, it's good enough. Thank you.
It's not your level. A student asked a question, right? See, it's random. It's random where this other X chromosomes, or which chromosome, whether it is from dad or mom, which one will become Barr body, and in which cell. It is completely random. Like a group of cells which gave rise to skin might have condensed X chromosome from a dad. A group of cells which gave rise to eye might have condensed X chromosome from mom, and dad might have given actually colorblind. Okay, so it's a little extra, but then a student asked, Professor, can that happen that the person got like this, a combination? So it is little extra. You might not, it's not required. If you think to that level, then I will address it. Continue.
Okay, number 10. Number 10, Calliope cat, also called tortoise shell cat, has black and orange patches due to Barr body formation, which is true.
This is exactly what I was talking about.
And the number 11, Morgan's D-hybrid cross explains why a pair of characters commonly appear together, which is true.
Mm-hmm.
And the number 12, commonly appearing pairs of characters sometimes separate and appear in uncommon combinations due to crossing over.
Yes.
Yeah, and the number 13.
I'm quickly checking, is it recording? Does it show for you?
Yes.
Okay.
Yeah, number 13.
Real quick, Tracy, for question number 11, he not only proved that pairs of characters commonly appear together and about linked genes, but he also was able to hypothesize that it is due to crossing over. So that was his contribution. And earlier I talked about two scientists completely opposing each other's viewpoints. So it is this, Morgan and Mendel. Morgan was a true scientist. He was an embryologist. He was a PhD. He was a scientist in a laboratory in modern times, right? Whereas Mendel was from 1857, something like that. So, and Mendel was a monk. Morgan is the one who ridiculed Mendel's work, saying that it was not done using scientific method. It is not even science. But then later on, whatever work he did, irony is that whatever he did was actually approving of what Mendel said. So that's a funny thing, how you should not go ahead and blurt out and insult someone's work without understanding. Always you should say that hey, there is a possibility that the person has not used the scientific method, so probably it is not 100% correct, or we cannot rely on that, and we should go ahead and further do the research in order to prove it. Yeah. Next.
Number 13, 4 types of changes in chromosomes are, the first one is deletion. The definition is remove a chromosome segment like A, B, C, D, and then you only have A, B, D. So C is missing. That's the deletion. And the second one is duplication. It's repeat segments like A, B, C, D, and then you have B, C again. And the third one is inversion. Inversion is reverse a segment within a chromosome, like A, B, C, D, and you got A, C, B, D. And the third one is translocation. Move segment from one chromosome to another chromosome. So there are 2 chromosomes, A, B, C, D, and the second one is like say X, Y, Z, and then you have X, Y, Z, A, B. And the first one missing AB, so that's translocation. And the number 4, oh yeah.
What was the answer for 12?
12, I wrote crossing over.
Yeah, good. And the changes in chromosomes, these are called chromosomal mutations or chromosomal aberration. A part of chromosome completely goes missing, that is deletion. A part is replicated and reattached, that is duplication. Inversion, it will go from left to right to right to left. So that kind of flipping on a chromosome, that is inversion. And translocation from one chromosome to another chromosome. This is the one which is important. Translocation is chromosomal mutation where the transfer of a part of a chromosome happens between 2 different chromosomes, not in the same chromosome, but between different chromosomes. Continues, Tracy.
Number 14, syndrome is genetic disorders. A set of traits are affected by this disorder. That's syndrome, like Down syndrome. And the symptoms is just like you feel what is wrong. So it's not a disease at all. So
I say have a headache. That's a symptom. I say have body aches. that is a symptom, right? So many symptoms put together is a syndrome. Actually, when you go to microbiology, you will see if you can say whether body aches is a symptom or sign. There is a difference between symptom and sign. Sign is something that you can measure. Can I, if Tracy says I have a headache, can I measure? It could be that, you know, she's just using an excuse, right? So symptom in here is both something that you can see and something that you can't measure. Altogether is symptom. And syndrome is a collection of many of them. Syndrome is a collection of many of them. Okay. Continue, Trace.
Okay, number 15. Down syndrome results from chromosome number 21. Is trisomic. So we should be pair of chromosomes, only 2, but for this one there are 3. Yeah.
Chromosome number 21. There are trisomy of 21. Those who did not fill up, listen to this, okay? So people who have Down syndrome, in every cell in their body, they have an extra 21. Not just 121 from mom and 121 from dad, but an extra 21 either from dad or mom. Either one, okay? It's mostly they say mom. So now the question for you is, in people with Down syndrome, in each one of their cell, how many chromosomes do they have?
46.
Number of chromosomes. Huh?
46 for human.
That's what I'm trying to say.
Okay.
They have one extra 21.
So 27 in total. So in total, Down symptoms.
Say the number correctly, Tracy.
It's 63.
What?
Isn't it? You just said extra 21, so 20.
Generally, okay, wait up. Generally, how many chromosomes in each cell?
46.
In human?
46.
46. I'm saying there is one extra. So how many?
47.
You said 27 earlier, Tracy. You wanted to say 47, but you said 27. Did you hear that, ladies?
Sorry, 47.
47. Stacy, 47, okay? 47. Just one extra. One extra, okay? So imagine in every single cell of their body, having one extra chromosome means instead of having 221 chromosomes with their genes, they have one just extra chromosome, and these genes are extra. That's all. That's all. A tiny molecule in the center of each of their cell with some extra genes is making them look so different, and their learning skill is different. The feelings are different. Can you imagine, like what such a tiny invisible molecule can do to a person, to a being, right? So that important the genes are. So we can't underestimate that a tiny molecule is extra. Big deal. No, forget about a tiny molecule. Even one molecule will make a difference. That is sickle cell disease. I hope you understood the intensity of what I'm trying to say. That doesn't mean that, oh my God, I can't even have 1 grain of salt extra. Not like that. We have something called homeostasis in our body, which takes care. But then when it comes to genes, yeah, we can't tamper with that. That is way too crucial than what we can estimate or what we can anticipate or think. Next.
The last one, Philadelphia chromosome is chromosome number 22 that has had a translation with chromosome number 9. So 22 will be shorter.
Understand, guys, this is Down syndrome is 21 and Philadelphia is 22. Make sure that you don't mix them because they're so close. The numbers are so close. You have to remember it really good. And remember, and look at this. Trisomy means 3. Trisomy means 3. So many times I've seen students mixing up 21, 22, 23, all in a blender. Okay, so be very careful. Down syndrome 21, Down syndrome 21, memorize like that. And if possible, like say it loudly, scream in the air or do whatever, I don't know. And Philadelphia 22-9, 22-9, 22-9. Maybe you can remember something like one is even, the other one is odd. Okay. Brittany, would you like to take over if you don't mind, please? We will give Tracy some rest.
You want me to do 17? I don't know. Did
16.
anyone else? You want me to do 16?
16. Yeah.
Okay.
Yeah.
Okay.
I'll do 17, not a problem.
Okay. So I have Frederick Griffith was attempting to prepare the vaccine for pneumonia and he discovered transformation.
Continue. Good.
And 2, Avery was the first scientist to provide evidence that DNA is the genetic material.
Okay, so there are many organic molecules, right? There is B vitamin in us, there are oils in us, omegas in us, neurotransmitters. Forget about that. Let's go a little bit higher in the sense, chapter 5. There are 4 kinds of organic molecules in our body. We are made of mainly 4 types of organic molecules. We call them CLPN, not CHON. Those are atoms. CHON is atoms. I'm talking about CLPN. Does anyone remember what is CLPN?
Carbohydrates, lipids, proteins, and nucleic acid.
So we are made of these. So they wanted to know whether genes are made of carbohydrates or proteins or lipids or nucleic acid. They did an experiment. Avery and his group did an experiment to show that no, they are not made of carbohydrates. No, they are not made of proteins. No, they are not made of lipids. Yes, they are made of nucleic acid. They did this experiment, all 4 together. Very smart experiment, really smart way to understand what things are made of, and they proved it. The reason why I'm talking about this is because this is a very important experiment. It requires you to think like a scientist, and it requires you to really understand the logic. And there could be a question on this on the test, and it's not easy to understand. Give it a couple of attempts at least, readings, when you read chapters 16 to understand that experiment. Next, Brittany.
State 2 of Chargaff's rules. I have base composition DNA varies between species, and any species is A equals number of T's and G equals numbers of C's. Okay.
Next.
Okay. And then
read again.
So 1 was base composition DNA varies between species, and then any species A equal number of T's and G equals numbers of C's.
Okay, so what they're trying to say from 1 is if you take all A's and T's and G's and C's and get a number for humans, okay, A might be all 35%. Okay, all A's are 35% in humans, whereas in chimpanzees, it could be 34.98%. That bit of a difference is there. Oh, really, that makes a difference? Yes, really, that makes the difference. That is what base composition means. Okay. Amount of sugar in lemonade, amount of sugar in your coffee. Isn't it different? It is different. Amount of sourness in carrot and amount of sourness in lemon juice. Isn't it different? Oh, H lot of different, right? I mean, heck lot of different, right? So that is base composition. If you're trying to understand base composition and you don't understand, don't brush it off. Please understand this is base composition. How much? Basically, how much is base composition? Next.
ATGCs are nitrogenous bases which become part of nucleotide. So a nucleotide is made of sugar, phosphate, and nitrogenous base.
Exactly. So do this again.
ATGC.
No,
Sorry.
no, I meant visually. Do this. Right hand in the front, left hand on the side. Right hand in the front, left hand on the side. Right hand is phosphate. Right hand is?
Phosphate.
And your body is sugar. Body is?
Sugar.
Left hand is nitrogenous base. Left hand is nitrogenous base. And they are of 4 types. What are they?
ATGC.
Adenine. Look at
Thymine.
my hand. Adenine, guanine, cytosine, thymine. Okay, it quite represents, whereas phosphate is same. So only this changed. Look, now this is adenine nucleotide. Everything else remains same except this change, right? So this is different. T, G, C. You see how it is different? So this entire thing changes just because this tiny part changed. I could make pancakes with salt, or I could make pancakes with blended cranberries. And it gives a different taste altogether, right? Or I could make usual pancakes with sugar. So that's what a nucleotide is. So don't get mixed up between the word nucleotide and nitrogenous base. Let me ask you this question. Which one is a bigger molecule between nucleotide and the nitrogenous base? Which one is a bigger molecule?
I wrote this down.
Is nucleotide bigger or nitrogenous base bigger? Okay, look at me again. You said nucleotide is this, right? Entire thing, right? This is nucleotide. And only this much is nitrogenous base. Only this much is nitrogenous base. So which is a bigger molecule?
The
You're
nucleotide.
muted.
The nucleotide.
Nucleotide is a bigger molecule. But if I do not ask this and you do not focus on it, you are mixing them up. And I'm asking you something and you're doing something else there because of that. But this entire thing, is it called A? Yes, it is called A because this is the one H which is changing. So I'm calling this entire thing by this name. Yeah. Okay. It's. I think I can give you a very good example. Doesn't matter whether it is Barack Obama or Michelle Obama or their girls, but they are called Obamas. As soon as I say Obama, who comes to your mind? Probably Barack Obama, right? Not Michelle Obama, right? It's like that. So this entire one's identity, it is determined or differentiated by this part. Okay, that is why we get mixed up between them. Next, Brittany, you're on mute.
So number 5, Rosalind Franklin is the unseen hero who produced the picture of the DNA molecule using the X-ray crystallography technique.
X-ray crystallography technique. Very good. She's the one who actually did all the experiments and the lab work, collecting the DNA, exposing it to X-rays. And just like you get a person from X-ray crystallography, all the bones, skeleton looks white and the other part looks black. Like that, you can get a picture of a DNA molecule, and that's what she did. X-ray crystallography is no different from human X-ray. Okay. But then she did it with a molecule, so she got a picture of the molecule inside structure, how the atoms are organized. She did all the work, but she never got the credit because she died before that work was given Nobel Prize, and the Nobel Prize was given to her boss, who is Maurice Wilkins, and Watson and Crick. James Watson and Francis Crick. They did not do the lab work, but then they got the Nobel Prize and the credit. Unfortunately, she never even got the credit. The only way that she gets the credit, it's not that she would care, but it's important to know who did it correctly, right? Because she was a woman those days. Women's work was not given importance. Yes, their work was subdued and their work was suppressed. Their identity or their role was suppressed. Those were the times in America too. Can you imagine? Yeah, in UK and America and many countries actually did not give importance to women's scientific work. That's a sad thing, and that can change only if we give importance to this kind of people who started doing this kind of work. Can you name any other woman scientists from olden times, ancient times? Not very ancient. Madame Curie. Madame Curie is another woman scientist who invented, I think, radium or uranium, one of them. Radium, I believe. Look it up. And she died of cancer because she didn't know that it is radioactive. She used to put it in her pockets and go around to do the experiments, and all that radiation actually gave her cancer and she died. Yeah, that's why that's why it's very important that you don't eat gum and do stuff like that in lab, because we don't know everything. Yeah. Yeah, there's so many things I say. I don't explain the reason, but there is a reason for so many things, and I don't have time in lab to explain all that. I just have to say, hey, this is lab rule, just do it. Okay. Okay, let's get to it.
All right, number 6 is describe these 4 properties of DNA in your own words. So I did double-stranded as 2 ropes.
Yeah.
And then I did helical.
1 second, ladies. 1 second. Give me a second, Brittany. Let me pause the recording. Where is the recording? So we were talking about Rosalind Franklin. That she is an unseen hero. That means she did not get the credit for the work that she actually did. She was the one who actually said that DNA molecule is double helix. But then who got the Nobel Prize were 3 other guys. The reason why she did not get any money, because she's done, she passed away. It is the phrase is Nobel Prize is not given posthumously. Means after the person is dead, Nobel Prize is not given. Understand that it is prize, means money, a lot of money. They want to give that money for the science research that they will do going forward. That is what they want to give money for. They do not give money like, hey, you did great, wonderful, take this money, enjoy. No, it's not for that purpose. Okay. Continue, Brittany.
Okay, so I put double-stranded as 2 ropes, and then helical as spiraled, like a screw thread or coiled.
Yeah.
And then I put anti-parallel is side by side running opposite directions.
Yeah,
And
it's exactly like traffic on either side of the yellow line. Sorry, I cut you. Traffic on either side of the yellow line. They have same distance between them, but only thing is if one is going towards north, the other one is going towards south. That is how it is, right? It is different in the sense in their direction. Next.
And then semi-conservative replication is half saving, making one old to one new. That's what I put. And then number 7 is nucleotides in a DNA strand are bonded to each other by covalent bonds. And then
Yeah. So look at this. These are the two strands, right? This is a polynucleotide strand. This is a polynucleotide strand in a DNA. Two together is a DNA. This is a polynucleotide. This is a polynucleotide strand. So this is a nucleotide. This is a nucleotide. This nucleotide to this nucleotide, the bond is covalent. So the bond between nucleotides in one strand is covalent bond. Nucleotides in a DNA strand, that A is important there. A DNA strand, A. Whereas, look at the next one. Go ahead, read, Brittany.
nitrogenous bases of two strands in DNA are bonded by hydrogen bonds between complementary bases.
So you look at this, the gap between my hands. This is hydrogen bonds. This is not a gap. They are bonded, but by hydrogen bonds, which is a weak bond. So it's like zipper sides. They can be easily ripped apart. They can be easily, not so easily. It does require some time because zip does not go on its own. You have to put in little tiny energy to unzip it, right? It's like that. So the bond between the nitrogenous bases of the two strands of DNA is hydrogen bond. So know the difference. Between is hydrogen and between nucleotides in the same strand is covalent, which is a stronger bond. Super important. So here is where you need to really pay attention. Okay, you have to really see what I want you to see. But how will I know? I'm saying it, but how will you know on the test? I'm not there to voice it out. Hey, see what I want you to see. What I want you to see is like so stupid to say. So I can't do that. But I did it. How? I bolded this. You see, it is bold for a reason. So if there is something like this, that means it is asking you to pay attention to that part. When I say pay attention to that part, I don't mean to say that you were distracted all the time. What I mean to say is, read that part couple of times. Think about it. Not only read, think about that part so that you can answer it correctly. I could do only this much, guys. I don't know what else to do on top of this, right? Okay. Brittany, next.
I have a quick question. So when it's talking about nitrogenous bases, is it talking about nucleosides with an S? Is that the difference? Or is that, am I thinking something else?
I wish I had two people here. I needed two people to explain that. Nitrogenous base is without a phosphate, I think. No, nitrogenous nucleoside versus nucleotide. Nucleoside is without phosphate. Nucleotide is with phosphate. So look at me again. Jocelyn, what are we going over? Okay, so nucleotide has three parts, right? A phosphate, sugar, and a nitrogenous base, right? So three parts is a nucleotide, excluding phosphate. Just don't put this, then it becomes nucleoside phosphate. what we are talking about in the activity sheet is nitrogenous bases, which is this of this nucleotide and this of this nucleotide. Between this and this of opposite strands is what bond between two strands.
Is
Is
hydrogen.
it clear, Tracy?
Yeah.
Okay.
Somebody answer Jocelyn's question, guys. Okay, continue, Brittany.
Okay, number 9, bacteria have circular chromosomes, whereas eukaryotes have linear chromosomes.
Yes.
Number 10, helicase proteins keep the unwound DNA strands separated from each other. Number 11 is RNA primer is synthesized during DNA replication by RNA polymerase.
No,
Oh, okay.
no. RNA primer is synthesized during DNA replication by primase.
Oh.
RNA is synthesized during transcription by RNA polymerase. RNA primer is synthesized during DNA replication by primase. There is a very little difference between the words, but it is there. It is actually clear. It is actually loud because we are saying RNA primer.
Okay.
But then if you focus only on RNA, then you will jump to the answer.
Okay.
DNA replication has no RNA polymerase. You might want to write that down. DNA replication has no RNA polymerase. DNA replication process has, for making RNA primer, the name of the enzyme primase is primase.
Okay. Do you want me to go
Tracy, what are you asking? Question number 8 by what?
Me?
You texted in the chat.
No, number 8 just answer question. Mariana already answered my question. Thank you, Professor.
Okay. Continue, Brittany, if you're okay.
Number 12, DNA polymerase cannot initiate DNA synthesis, and that's true.
Mhm.
DNA polymerase can add nucleotides to an existing RNA or DNA strand, 3' and only. I put true.
Again, one more time, I mentioned this earlier in some of the videos. Whenever I give statements like this, they happen to be true, most of them, not all, most of them. And why did I give true statements? And again, on top of it, why am I revealing that, right? Why am I disclosing it? It's because I want you to know that I want you to memorize these sentences. These are the basic fundamentals on which many things will be built. So you have to sit and memorize this entire sentence. It is required. It is essential. Continue, Brittany.
Okay. 14, DNA polymerase not only synthesizes new DNA, but also carries out mismatch repair. That one's true. RNA polymerase synthesizes RNA primer. True.
Huh? I just said.
Oh, that's DNA. Okay.
What synthesizes RNA primer?
The primase.
Primase.
Yeah, I mentioned it. It is just a different way of phrasing the question. Point number 11 was a blank, and point number 15 is a true or false question, and this happens to be false because RNA polymerase does not synthesize RNA primer. Primase enzyme synthesizes RNA primer. So this is false. I gave this so that you can learn, not because I get any extra pleasure by doing this, okay? Yes,
I got that one. I got that one wrong because I already had that one wrong.
Yes, Tracy.
Professor, I have a question about number 12. Number 12, DNA polymerase cannot initiate DNA synthesis, which is true. So my question is, what initiate DNA synthesis?
Primer.
Okay.
The primer is already there in place, and then DNA polymerase will come and add DNA nucleotides to it.
So
Okay.
that primer is the beginning. Okay.
Primase will start RNA primer. When RNA primer is in place, then DNA polymerase will come and add nucleotides to the 3' end of it. And I always say this example, like for example, Lady Gaga has to perform. Okay, does she come to the place where she has to perform, and does she do all the decorations and putting out the chairs or whatever it is, setting up the sound system? She doesn't do, right? She won't do any of it. Actually, she doesn't do even her makeup, her own dress she doesn't wear. Somebody puts it on her. You all know this, right? So I say that DNA polymerase is like that. It's a celebrity. It's like Lady Gaga. I always say this name, Justin Bieber. So DNA polymerase is our celebrity. They don't come and they don't do the arrangements. They come and just perform. Right? So that is how it is. So DNA polymerase will synthesize new DNA, but it will not start. Somebody else has started, then DNA polymerase will come and add. Good. Next.
Number 16, lagging strand requires multiple RNA primers, and this is true. A lagging strand is made of Okazaki fragments. And then leading strand is synthesized continuously, and lagging strand is synthesized in fragments.
Discontinuously.
Okay.
It's the same thing, but then it just flows together very well, because here you said continuously. The second blank, you will say discontinuously. It's just easy to memorize.
Okay. 19, two types of DNA repair are mismatch repair and nucleotide excision repair. And then number 20 is histones are the proteins that help in packaging of DNA. And then 21, our nucleosome is made of chromatin and histone proteins.
Nucleosome and histone protein.
I put DNA and histone protein.
Yeah, that's right.
DNA.
That, Brittany, would you please change it? DNA and histone proteins. Okay. Because nucleosome is just one round, one circled part of combination of protein, histone, and DNA. That is nucleosome, right? So it's actually this. Next. Oh, we are done?
Yep.
Oh, by the way, real quick, chromosomal aberrations, this table is there in the textbook or in my PowerPoint. You can find it easily for the explanation. And same thing, different types of RNA and their function. It is the first slide in my first few slides. One of them is this table in my PowerPoint for chapter 17. Am I recording?
Yes.
Okay. Anybody else who has answers for chapter 17 activity sheet? Brittany, Stacy, do we have another Wednesday session where we will meet before your quiz 5A and 5B?
No.
This is the last one?
Yep.
How long can you go on? Can you put it in the chat? I had recorded that. I wish I had recorded that part. It was a nice thing to know for people. Anyway, continue, Brittany.
Okay, do you want me to start at 1 or 2?
Where were we? One is done. You don't need to. Everybody can look up. It's table is given
Oh,
in the
Professor,
PowerPoint.
no, Professor, I have a question about number 1, because I have mRNA, tRNA, RNA. I'm missing 4 and 5. Is it pre-RNA and snRNA? I can look up their functions, but I need the names.
Primary transcript is what I have, and then small nuclear RNA or snRNA.
Yes,
Pretty, you
small
got
nucle-
it from the PowerPoint, right? PowerPoint initial slides, it is there, Tracy. It is siRNA and short interfering RNA and sRNA, small RNA. Short interfering RNA and sRNA. It could be primary transcript. It cannot be primary transcript. No, it is not primary transcript. Don't take that, Briti. Is that what is given in the list in the PowerPoint?
I can't remember.
Okay, I'll get back to that question a little towards the end. But here are the 5: mRNA, rRNA, tRNA, siRNA, and I believe the other one is and the
Pre?
5th one even I'm
It's pre-RNA,
tRNA. No, there is no. It's,
pre-mRNA, no?
no, it's not. We did research on that. I'll get back to this little towards the end. We have a couple more minutes. In fact, let's pause it here. Why don't you Google it? You will definitely find the different types of RNA, and I'll get back. Okay, I'll pause the recording. You guys stay. Don't end. I'll be back. Recording. So there are 5 types of RNA. mRNA for messenger RNA, rRNA for ribosomal RNA, tRNA for transfer RNA, siRNA for short interfering RNA or small RNA, and the 5th one is miRNA, which is microRNA,
MI?
MI. Like microbiology, MI, yes.
So the small nuclear RNA is not one. It's siRNA, short interfering.
Or small interfering RNA. siRNA is short interfering RNA or small interfering RNA. Look at this. This is the list.
Don't let it go
Okay.
away. Hold on.
Is there any way I can put the link in the chat and you
So,
can click on it and see?
So, Professor, it's not small nuclear RNA. It's not that one?
snRNA also. Yeah, that is another one. Small nuclear RNA. snRNA and siRNA are very similar, very similar.
Okay.
You can consider siRNA and snRNA together. I would like to take it as 5 types of RNA instead of 6 types of RNA. Next.
Okay, number 2 is gene expression is a process by which DNA ultimately directs protein synthesis.
Yeah, DNA protein synthesis.
Mhm.
Yes.
And then number 3, gene expression includes 2 stages, transcription and translation. Transcription is the synthesis of RNA using information in the DNA or nucleus. And then
Transcription is the synthesis of RNA using information in the DNA.
Okay.
So it's
And
And
MR.
it could be any of these 5 types of RNA. So
Oh,
transcription is not just mRNA. Transcription could be any of these RNA, 5 types.
okay. And
Was
then
that your question, Tracy?
yes, because I wrote mRNA, but looks like it's RNA for all types of RNA. Got you. Thank you.
All types. See, I read minds. Stacy, I not only teach science, I also read minds. There is a name for them. I'll let you guys look up.
My mind is easy to read. Yeah.
My mind is easy to read. All right. Okay, then read my mind. Okay, Brittany, go on.
translation is the synthesis of polypeptide using information from mRNA.
That's so right. That's so right. It is synthesis of a polypeptide from mRNA, not any other RNA, because only mRNA can be translated. All other RNAs are not translated. There are 5 types of RNA, but other RNAs are not translated. Only mRNA will go through translation. And when an mRNA is translated, what is made? A polypeptide is made, not protein, a polypeptide. What is the difference between polypeptide and protein? A protein could be only one polypeptide, or a protein could be 2 or more polypeptides. That means these 2 or more polypeptides came from different mRNAs? Yes, it is possible. One more time. A protein is either one polypeptide or more than one polypeptide. A polypeptide is made from only one mRNA. Another polypeptide is made from another mRNA. Third polypeptide is made from another mRNA. They are all put together, can be a protein. So a protein can be one polypeptide or many polypeptides. One polypeptide example is insulin. One polypeptide equals one protein is insulin. 4 polypeptides is one protein is hemoglobin. It's not actually heme part. Heme is iron. Okay. Continue, Brittany.
Sorry. Number 6, transcription takes place in nucleus in eukaryotes and cytoplasm in prokaryotes.
Good.
Okay. And then translation takes place on ribosomes in the cell cytoplasm. And then number 8, only in eukaryotes there is an intermediate stage between transcription and translation called RNA processing.
Good.
Okay. And then a codon has 3 nucleotides, hence it is called a triplet code.
Yeah, triplet, like triplets. 3 kids born at the same time, like that. Triplet code. Next.
The end product of transcription is RNA.
Or any RNA, right? Any RNA.
Okay.
Or all types of RNAs are formed by transcription only, the process. Is mRNA formed at the end of transcription? Yes. Can rRNA be formed at the end of transcription? Yes. Next.
Okay. The end product of translation is a polypeptide.
A polypeptide. Good. Next.
DNA template strand is complementary to code strand and mRNA. I put true. And then mRNA.
Wait. I need time to process.
Oh, sorry.
Coding strand and mRNA.
Professor, what is
Complementary to
coding
coding
strand?
strand.
What is coding
One
strand?
second. It is.
Yeah.
In DNA, there are 2 strands. Look at my hand. In DNA, there are 2 strands, right? This is one is called template. The other one is called coding. They're complementary to each other. That means if there is a A here, there is a T here. If there is a G here, there is a C here. They're complementary, like Lego pieces. They fit in well, right? Like that, they're complementary. So the one which is gene, with where the gene is, it is called template strand. And the template strand is used for making mRNA, RNA nucleotides. So template is complementary to mRNA. Template is complementary to mRNA.
Mhm.
So this coding strand is complementary to template. Because it is complementary to template, it is also complementary to mRNA. It's hard to explain like this. You have to watch the video.
12
You have
times.
to go over the entire concept. But the answer is true for now. Next.
Okay, mRNA strand is similar, not same to the coding strand. And I
Yeah.
put and then mRNA have
True.
T. Yes, I put true.
13 is true. That the emphasis is on is similar, not same. Because in mRNA, there are U's, whereas in DNA, there are T's. Excuse me. Next.
14 mRNAs have T's in the strand. No. The answer is no.
mRNAs do not have T's, means thymines. They have
Uracil? U.
uracil. Yeah, U. Next.
Okay, direction of replication, transcription, and translation is 5' to 3'. And I put true.
True. And answer for 14 is no. Question is saying mRNA can have T's in the strand. Answer is no for 14. 15 is true. Next. 16.
Okay. 16, start codon AUG codes for methionine amino acid.
Methionine, I think. Yeah, methionine.
Let me type it.
I typed it here.
Oh, okay. And then stop codons code for 0 amino acids.
Yeah, 0. None. 0 means none.
And mRNA has 30 nucleotides, including a stop codon. The polypeptide it codes for has 10 amino acids.
No.
Okay.
See, 30 includes a stop codon. Right? So 30 minus 3 will be 27. 27 divided by 3 will be? What is 27 divided by 3, ladies? 9.
9.
And cell phone. Ladies and cell phone. 9 is the answer for 18.
Okay. Oh, okay. 19, an RNA polymerase starts transcription at initiation and ends at termination of a gene.
You're right.
Okay. In
Promoter, sorry. 19, and RNA polymerase starts transcription at, wait, RNA polymerase binds to promoter, but starts at initiation. You're right. No, initiation. What are the 3 parts of a gene? Am I recording this? Is recording going on? I can't see. Yeah.
Yes, it's recording. Okay.
So the answer for that was a promoter, right? Okay. RNA polymerase starts transcription at promoter and ends at terminator, like your Terminator movie. Yeah, terminates. means ends. Next.
Okay. And then in eukaryotes, transcription factor proteins mediate the binding of RNA polymerase.
Yeah, transcription factor proteins. I mentioned this many times that I have a skin color gene in my red blood cells. But are my red blood cells going to make my skin color? I also have hemoglobin gene in my skin cells. Am I making hemoglobin or insulin here? You have insulin gene in my skin cells. Do my skin cells make insulin? No, they don't. If we had made like that, we would have never had diabetes. And it will be too much. Actually, it will result in another disease if you were doing that. Next.
I don't have a very good answer for 21.
Difference between RNA splicing and alternative RNA splicing is RNA splicing is removing introns and keeping exons because exons are expressed. When we select which introns are removed but not remove all introns, then it is called alternative RNA splicing. The best example is this. Every year, or not every year, very frequently the flu virus is changing, right? Flu virus is changing, but then you will not go get a flu vaccine for every variant for a flu. Your body makes different antibodies for different variations of flu. It does this by taking your antibody mRNA and making different polypeptides with the same mRNA, with the same genes. Flu virus and so many viruses and bacteria are changing and mutating out there. Do we get every year new genes? Do we buy it from Publix and pop in? No, we can't, right? Whatever we have, we are born with a set of genes, we are done. We're done. If we have the immunity because of that, we are great. If we do not have, then we are doomed. Right? That is what happened during COVID, basically. So that is a good example. Alternative RNA splicing is a good example for different antibodies we happen to make using the same gene pool that we have. That is called alternative RNA splicing. Selecting which introns will be removed. Not all introns are removed. That is alternative RNA splicing. Do you have the next one, Brittany?
Function of amino axle T
Amino acyl.
amino acyl, is that right?
Mm.
tRNA synthetase is to translate tRNA.
No. Charged tRNA means adding an amino acid to tRNA. We call it like charging your cell phones. Adding an amino acid to tRNA. It's called charged tRNA.
Okay.
No worries. Thank you, though. Next.
Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons.
Very good. tRNAs have anticodons. They bind to the codons of mRNA. Very nice. Yeah.
Okay, and then E site is the exit site on ribosome.
Next.
3 steps in translation elongation are codon recognition, peptide bond formation, and translocation.
Translation elongation 3 steps. I have to check this. Give me a sec. So if they ask a question like what are the 3 stages of translation, then we would say 3 stages of translation are initiation, elongation, and termination. if they ask what are the 3 steps in translation elongation, we would use this. 3 steps in translation elongation are codon recognition, peptide bond formation, and translocation. I'll stop sharing this and then I'll share the other question. Ribosomes facilitate, ribosomes facilitate specific coupling of the E's exit site. 24 E's exit sites. 3 steps in translation elongation are the ones that, can you repeat the 3 steps of elongation, translation elongation, Brittany?
Codon recognition, peptide bond formation, and translocation.
Wonderful. And that is a process. I do not want to get into it. Make sure that you watch the video, guys, of translation elongation. If it is not done in my videos, make sure that you go to YouTube and watch translation elongation process. Okay, next.
Insertion or deletion of nucleotides from any gene produce frame shift mutation.
Yeah, this is called frame shift mutation.
Can you repeat it?
This insertion.
Frame shift.
Frame shift mutation. So if I ask you what is my hand from here to here, you will say palm, right? From here to here is palm. Whereas I switch it, okay, and I say, what is this? Frame has shifted, right? Now you cannot say this is palm. This is part of my hand, or this is wrist. You will call this wrist. You'll call this palm. Like that, frame has shifted. So this is a frame. If this is the frame, you will say palm. If this is a frame, this is called wrist. So frame has shifted. Frame has shifted. That means when you read AGG, you'll put 1 amino acid. Whereas if A got deleted, then you will read GG, whatever is next, let's say GGC. And you'll put a different amino acid. That is what is happening, and that is what results in so many diseases. This is called frame shift mutation. I explain more in my videos for sure, or else there must be a reference lecture videos. By the way, this insertion and deletion are not same as chromosomal aberration. That is part of a chromosome, which is many nucleotides. Whereas this insertion and deletion mutations are just one nucleotide, one. That's why these are called point mutation types. These are point mutations. Okay, one new nucleotide got inserted somewhere, or one nucleotide got deleted from somewhere, and that results in a disease. Just one molecule can cause a disease. Next, Brittany.
27, I have nonsense mutation changes an amino acid codon into a stop codon.
Nonsense results in stop codon. It is there in my PowerPoint. Check and make sure. Next, mRNA codon AUA will pair up with
UA.
Guys, exactly. This is our RNA. This is also RNA. Okay, so A is complementary to U. U is complementary to A. A is complementary to U. So what you write here is UAU. Like this, I could have given anything. This is just an example. It's not that I'll give AUA only on the test. Next.
How are the instructions for assembling amino acid into protein encoded into DNA? Use 3' AATA CGG CAAA ACT 5' DNA sequence for writing an mRNA sequence.
We will pause here. It is 11:02. Those two are the questions that I was talking about. It needs a lot of explanation. And I'm wondering if there is already a video for that, protein synthesis video. Did I make a video for that already? Let's
Professor, we can't hear you.
You're not able to hear at all? Now you're able to hear?
You're good now.
We're good now? So what I'm going to do is I'm going to record questions 29 and 30 from chapter 17 activity sheet separately. I'll stop this recording.